Welcome to this comprehensive lesson on neurons, the fundamental units of the nervous system. In this module, we will explore 5 critical aspects of neuronal function. First, we will understand what neurons are, their unique characteristics, and the fascinating history of their discovery from the pioneering work of Golgi and Cajal to the groundbreaking research of Hodgkin and Huxley. Second, we will examine the detailed structure of neurons, learning about dendrites, soma, axons, myelin sheaths, and synapses, and how each component contributes to neuronal function. Third, we will classify neurons by their structure, function, and the neurotransmitters they use, understanding why different types exist and where they are located in the body. Fourth, we will dive into the resting membrane potential, discovering how neurons maintain an electrical charge difference across their membrane through ion gradients and the sodium-potassium pump. Finally, we will explore action potentials, the electrical impulses that allow neurons to transmit information rapidly throughout the nervous system. By the end of this lesson, you will have a complete understanding of how neurons work, from their molecular machinery to their role in creating thought, sensation, and movement. Main content. Let us begin with the core definition. A neuron is a specialized, electrically excitable cell that transmits information throughout the nervous system via electrical impulses and chemical signals. Neurons possess 5 key characteristics that make them unique. First, excitability. Neurons can generate electrical signals, called action potentials, in response to stimuli. Second, connectivity. A single neuron can form synaptic connections with 10,000 or more other neurons. Third, polarity. Information flows in one direction through a neuron, from dendrites to cell body to axon to synapse. Fourth, postmitotic. Most neurons cannot divide or regenerate after development. Fifth, plasticity. Neurons can strengthen or weaken their connections based on activity patterns. Historical timeline. Now let us journey through the remarkable history of neuronal discovery. 1873, Camillo Golgi and silver staining. Italian physician Camillo Golgi developed a revolutionary silver chromate staining technique that allowed scientists to visualize individual neurons for the first time. 1891, Santiago Ramón y Cajal and the Neuron doctrine. Spanish neuroanatomist Santiago Ramón y Cajal used Golgi's staining method to make a groundbreaking discovery. Neurons are discrete individual cells, not a continuous network. This became known as the Neuron doctrine. Golgi and Cajal shared the 1906 Nobel Prize. 1906, Charles Sherrington and the synapse. British neurophysiologist Charles Sherrington coined the term synapse to describe the junction between neurons. 1939, Hodgkin and Huxley action potential model. Alan Hodgkin and Andrew Huxley developed the first quantitative mathematical model explaining how action potentials are generated. 1952, Katz and Eccles on neurotransmission. Bernard Katz and John Eccles demonstrated that synaptic transmission is primarily chemical, not electrical. Neuron structure and anatomical components. The neuron you see illustrated here is a multipolar neuron. Cell body, soma. First, the soma, or cell body. This is the metabolic center of the neuron. It contains the nucleus with the neuron DNA, Nissl bodies for protein synthesis, mitochondria for energy production, and Golgi apparatus for packaging proteins. Dendrites, extending from the soma, are the dendrites, highly branched projections that serve as the neuron input zone. They receive incoming signals from other neurons. Axon hillock. At the junction between the soma and axon lies the axon hillock. This is where action potentials are initiated. If the membrane potential reaches threshold approximately -55 millivolts, it has an extremely high density of voltage-gated sodium channels. Axon. Extending from the axon hillock is the axon, a long, cylindrical projection that conducts electrical impulses away from the cell body. Myelin sheath. Many axons are wrapped in a myelin sheath. In the central nervous system, myelin is formed by oligodendrocytes. In the peripheral nervous system, myelin is formed by Schwann cells. Myelin increases conduction speed through saltatory conduction, nodes of Ranvier. Between myelin segments are gaps called nodes of Ranvier. These are unmyelinated gaps where ion exchange occurs. Axon terminals. At the end of the axon are the axon terminals. They contain synaptic vesicles filled with neurotransmitters. Synapse. Finally, the synapse, the junction between the neuron and its target cell. It enables intercellular communication through chemical or electrical signals. Types of neurons, classification systems. Classification by structure. 1 multipolar neurons, 1 axon and multiple dendrites. Most common type in the brain and spinal cord. 2 bipolar neurons, 1 axon and 1 dendrite, found in specialized sensory organs like the eye and nose. 3 unipolar neurons. A single process extends from the cell body, then splits into 2 branches. 4 anaxonic neurons. Multiple dendrite-like processes, but no identifiable axon. Classification by function. 1 sensory afferent neurons. Carry information from receptors to the central nervous system. Tau motor efferent neurons. Carry commands from the central nervous system to muscles and glands. 3 interneurons. Located entirely within the central nervous system. They make up approximately 90% of all neurons. Classification by neurotransmitter. Year glutamate-ergic neurons. Release glutamate, which is excitatory. 2 GABA-ergic neurons. Release GABA, which is inhibitory. 3 cholinergic neurons. Release acetylcholine. 4 dopaminergic neurons. Release dopamine. 5 serotonin-ergic neurons. Release serotonin. 6 noradrenergic neurons. Release norepinephrine. Resting membrane potential and ion gradients. Every neuron maintains an electrical charge difference called the resting membrane potential. This is approximately minus 70 millivolts. Ion distribution. Inside the cell, potassium concentration is high 140 millimolar, while sodium is low 12 millimolar. Outside the cell, sodium is high 145 millimolar and potassium is low 4 millimolar. Sodium-potassium pump. The sodium-potassium ATPase pump is the primary mechanism. It actively transports 3 sodium ions out and 2 potassium ions in. Potassium leak channels. These channels are always open, allowing potassium to diffuse out of the cell, making the interior negative. Nernst equation. The Nernst equation calculates the equilibrium potential. For potassium, it is minus 90 millivolts. For sodium, it is plus 60 millivolts. Goldman equation. The Goldman-Hodgkin-Katz equation calculates the actual resting potential. Because the membrane is most permeable to potassium, the resting potential is close to minus 90 but sits at minus 70 millivolts due to some sodium permeability. Action potential phases and ion channel dynamics. An action potential is a rapid all-or-nothing electrical signal. Phase 1, resting state. The neuron is at minus 70 millivolts. Voltage-gated channels are closed. Threshold. If a stimulus depolarizes the membrane to minus 55 millivolts, an action potential fires. Phase 2, depolarization. Voltage-gated sodium channels open. Sodium rushes into the cell. The membrane potential reaches plus 40 millivolts. Phase 3, repolarization. Sodium channels inactivate and voltage-gated, potassium channels open. Potassium rushes out of the cell. The potential falls back toward negative. Phase 4, after hyperpolarization. Potassium channels are slow to close. The membrane potential transiently drops to minus 80 millivolts before returning to minus 70. Refractory periods. During the absolute refractory period, no new action potential is possible. During the relative refractory period, a stronger than normal stimulus is required. Key properties. 1. All or nothing. 2. Non decremento. The signal does not weaken. 3. Unidirectional. 4. Frequency coded. Information is encoded in the rate of action potentials. Synaptic transmission. This diagram illustrates the 8 critical steps involved in chemical synaptic transmission, the process by which neurons communicate with each other. Step 1. Action potential arrives. Step 1. The action potential arrives. An electrical impulse called an action potential travels down the axon and reaches the presynaptic terminal, the endpoint of the sending neuron. Step 2. Calcium channels open. Step 2. Calcium channels open. The arrival of the action. Potential triggers voltage-gated calcium channels in the presynaptic membrane to open. Calcium ions rush into the presynaptic terminal. This calcium influx is the critical trigger for neurotransmitter release. Step 3. Vesicle fusion. Step 3. Vesicle fusion. The influx of calcium ions causes synaptic vesicles, tiny membrane-bound sacs filled with neurotransmitter molecules, to move toward and fuse with the presynaptic membrane. Step 4. Neurotransmitter release. Step 4. Neurotransmitter release by exocytosis. The fused vesicles release their neurotransmitter contents into the synaptic cleft, the tiny gap between the presynaptic and postsynaptic neurons. This process is called exocytosis. Step 5, diffusion across cleft. Step 5, diffusion. Across the synaptic cleft. Neurotransmitter. Molecules diffuse randomly across the synaptic cleft. This is a passive process driven by random molecular motion. The journey takes less than 1 millisecond. Step 6, receptor binding. Step 6, receptor binding. Neurotransmitters bind to specific receptor proteins embedded in the postsynaptic membrane. This binding is highly specific, like a key fitting into a lock. Glutamate binds to AMPA and NMDA receptors. GABA binds to GABA-A and GABA-B receptors. Step 7, ion channels open. Step 7, ion channels open. Receptor. Binding triggers ion channels to open in the postsynaptic membrane. This creates either an EPSP, which is excitatory, or an IPSP, which is inhibitory. Step 8, neurotransmitter removal. Step 8, neurotransmitter removal. To prevent continuous stimulation, neurotransmitters must be rapidly removed. This happens through reuptake, where they are pumped back, enzymatic degradation, or diffusion. Neurons do not work in isolation. They integrate signals from thousands of other neurons and adapt their connections based on experience. This is the foundation of learning and memory. Section 1, summation. Let us examine summation, how neurons combine multiple inputs. Spatial summation is when a single neuron receives inputs from many different presynaptic neurons simultaneously. Temporal. Summation is when a single presynaptic neuron fires repeatedly in rapid succession. If enough EPSP accumulate quickly, they reach the -55 millivolt threshold. Section 2, neural circuits. Now let us explore neural circuits. Divergence is when one neuron synapses onto many target neurons. Convergence is when many neurons synapse onto a single target neuron. Reverberation occurs when neurons are arranged in a circular pathway, creating a self-sustaining loop. Section 3, plasticity mechanisms. Finally, let us examine plasticity mechanisms. Long-term. Potentiation, or LTP, is when the connection between two neurons strengthens. Long-term depression, or LDD, is when the connection weakens. Structural plasticity is when neurons physically change their structure, like growing new dendritic spines. Neurogenesis is the formation of new neurons in the adult brain. Understanding neurons has profound real-world applications that affect medicine technology and our daily lives. Application 1, neurological disorders. When neuronal function goes wrong, devastating diseases result. Epilepsy involves hyperexcitability, where neurons fire excessively. Parkinson's disease results from dopamine depletion. Alzheimer's disease features amyloid plaques that disrupt synaptic transmission. Application 2, pharmacology. Most psychiatric medications target specific neuronal mechanisms. SSRI blocks serotonin transporters to treat depression. Benzodiazepines enhance GABA-A receptor function to increase inhibition. Local anesthetics like lidocaine block voltage-gated sodium channels to prevent action potentials. Application 3, brain-computer interface. Brain-computer interfaces, or BCI, represent a revolutionary technology. Electrodes implanted in the brain decode neuronal firing patterns into digital commands, allowing paralyzed patients to control prosthetic limbs with thought alone. Application 4, artificial neural networks. Artificial neural networks are computer systems inspired by biological neurons. These models use weighted connections mimicking synaptic strength to power image recognition and natural language processing. Application 5, neuroprosthetics. Neuroprosthetics use electrical stimulation to bypass damaged sensory pathways. Cochlear implants restore hearing by directly stimulating the auditory nerve. Let us consolidate our understanding of neurons by reviewing key concepts and correcting common misconceptions. Section 1, summary checklist. 1, neuron definition. Neurons are electrically excitable cells. 2, structure. Information flows from dendrites to soma to axon. 3, resting potential. The potential is -70 millivolts. 4, action potential. It is an all-or-nothing event. 5, propagation. Signals travel via saltatory conduction in myelinated axons. 6, synaptic transmission. Involves neurotransmitter release and receptor binding. 7, integration. Neurons sum excitatory and inhibitory inputs. 8, plasticity. Connections strengthen through LTP. 9, classification. Neurons are classified by structure and function. 10, clinical relevance. essential for treating neurological disorders. Section 2, Common Misconceptions. Misconception 1, neurons touch directly. Correction, neurons do not touch, they are separated by synaptic clefts. Misconception. 2, action potentials vary in strength. Correction. They are all or nothing and frequency coded. Misconception. 3, myelin insulates the entire axon. Correction. It forms segments with gaps called nodes of Ranvier. Misconception. 4, dendrites conduct action potentials. Correction, only axons conduct action potentials. Misconception. 5, neurotransmitters cross the membrane. Correction. They bind to receptors on the membrane, they do not enter the cell. Misconception. 6, all neurons fire at the same rate. Correction, firing rates vary dramatically by neuron type. Remember, neurons are the biological basis of thought, sensation, and action. Understanding neurons is understanding the essence of who you are. Closing.